be able to draw a short single-stranded polynucleotide, distinguishing between purines and pyrimidines, structural differences between the ribonucleotide versus a deoxy, proper Watson-Crick base pairing. That's really just at the letter, right? So it's good to be able to do the hydrogen bonding. I'm not really even pushing you that far. I hope you could recognize what could hydrogen bond, but if you were like, I missed one, I'm not going there. Just get it right. What is the expected matching pairs? Why does DNA exhibit a major minor groove, a flexible conformation of that? And really, this feeds into a lot of other things below that, but DNA is longer, okay? And RNA is shorter. That's really, and DNA likes to be double-stranded. Can RNA be double-stranded? Absolutely. But because RNA is usually shorter, we're giving it more opportunities in single-stranded form to fold back on itself and find structural other stability, right? 3 types of double-stranded DNA, features of RNA that allow more structural diversity, and you already know all of these structural motifs that contribute to the tertiary structure, important physical chemical properties, relevance of DNA-GC pairing for temperature, stability, etc. So this one is all about cloning, bacterial phases versus vectors, selection of DNA of interest, power changed of DNA sequencing. So GMOs we've already mentioned briefly. We're not going to go much more into that at all. PCR we want to talk about. Personalized medicine, next gen, we already mentioned that. So wow, lots of fun stuff to go through. Okay. Some of you have already done this. Is doing cloning, is cutting with a restriction endonuclease, is this like super rocket science? If your answer is yes, at what period of time? Because currently high school students are doing this. So stop feeling like this is outside your pay grade. If high school students are doing this, then, and you're a senior student, stop making excuses, get down to business and say, I can learn this, okay? Okay, so what we need to do, cut it, select a suitable carrier, a vector, insert our gene of interest into it, insert the recombinant vector into the host cell, and then produce a whole bunch of copies. In looking at this, if this is my gene of interest, here's the vector, we're saying a plasmid. Somebody help me out. When we say a plasmid, what are we really talking about? We're talking about a bacteria, aren't we? Bacteria have plasmids. They're inserted pieces of DNA that are sort of extra that they picked up that give them certain genetic advantages. They keep it along. How long? As long as it gives them a selective advantage, and then they sort of throw them away. They don't have to keep it. So if I give a bacteria a new plasmid with an interesting vector in it, and I don't put a selective pressure on it, what will it eventually do? It'll spit it out. It won't be the predominant one, because it's extra work to produce something and keep it around if it doesn't have any value. So we put a selective pressure on to keep it. Okay, so then I cut it open, and when I cut it open, I can insert my new piece. If I open it up, can it reclose on itself without having inserted the piece? Yes, it can. Is it possible then that I could put more than one of these in? Yes, it's true. I could put more than one copy in. But assuming that we got the right copy in and then I put it in here, do they always go right into the cell? No, there's a little trick to get them in, but they don't always work. So is it possible I get more than one copy or no copies? Yes, all of that is true. So considering all those options, here's some vocabulary for us, okay? Restriction endonuclease. Really two major forms you're looking at. This is a pair of DNA scissors. They come in ones that'll make sticky ends versus blunt ends. When I say a sticky end, We are talking about regions of the DNA that as I'm going along 3' to 5' in the double-stranded region, that I actually have a sticky piece. Meaning I've got a region that would overlap with another region when I put it in, that I'm going to get my pieces to connect, okay? So that's a sticky end. If I do a blunt end in my double-stranded DNA, I'm really just cutting it straight across. And that means that when I put them together, there is very little specificity to that region. So sticky ends give me more specificity when I'm re-adding something, but we use both, okay? There are companies like Rebase here, they specialize in giving you the restriction endonuclease of your choice to cut exactly where you want to cut it. That's their job. Make you find you some DNA scissors, that'll do its job, okay? It has to recognize the sequence. There's a short sequence it recognizes to cut. There are lots of them. But we typically use a whole bunch of plasmids which are well characterized for our purposes, so most of the stuff you're using is not a big deal. The next one's DNA ligase. This is literally DNA glue. Put them back together, okay? So there are enzymes that repair DNA typically, and they use ATP. Bacteria uses NAD+, and they put things back together for us, okay? When I'm looking at selection of the right plasmid to use, I could use just a regular one. What's the restriction here?
15,000.
About 15,000 is about the biggest we can get of inserting my new interesting piece. So that's actually pretty limited, just to be honest. We can get some things in there, but it's limited. If I want to use something like a back, an artificial chromosome is bigger, I can put more information in, sometimes up to 300,000 base pairs, so significantly bigger, right? We can use those in something like, so here, antibiotic resistant genes commonly used, they're circular. Over here, stable origins of replication for both of them. I could get into some nice yeast ones that we really like. What's the difference between yeast as a single-celled organism versus a bacteria? What's really the difference?
Well, the yeast is a eukaryotic cell, right?
So what advantage does that give me? More similar to humans because it's eukaryotic, but what advantage?
I mean, the systems that they have for reading and processing the proteins that they make are more similar to us. So like if we wanted to make a human protein.
So when we talked about proteins misfolding, we were talking about things like chaperones and chaperonins. The ones that will exist will be more similar to ours if we're talking about a eukaryotic system. What's it called when a piece of RNA gets all clumped up and it has a region that it is actually going to remove? An intron region, right? It's going to cut out, not even going to use. Does that exist in bacteria? Well, then what would I do with that if I had for example, a human gene, I'm throwing it in there and it's got a big intron in the middle of it? It's going to be a problem, right? Yeah.
So a question, not directly about that, but with actually like doing these techniques in the lab, right? Because like if you have a plasmid that you're putting into bacterium, right? It's pretty easy. Like you said, like I've done like heat shock insertion and stuff, and you heat them up and you make them uncomfortable. And like, oh crap, we're dying, and you put the plasmid in, and they will intake the plasmid, right? But when you are doing these larger like artificial chromosomes, when you're putting it in yeast or something, what kind of technique is used to actually do that?
Yeah, so the electroporation techniques are a little bit different. Yeah, we actually do a partial digest even of their membrane.
Oh, okay.
We put in pretty harsh conditions.
Use them thoroughly.
Yeah, so, yep, good points, okay? Again, blunt versus sticky ends, easily visualized there for your viewing pleasure, but what we just talked about, again, multiple repeats help us remember what we're dealing with. Okay. Anna, go ahead, Lee.
For the vectors, there was a question on the
Yeah.
study guide about bacteriophages versus.
We're gonna talk about that too.
Okay, later.
We're getting there. You're on track. Ampicillin is what? With that psyllin hanging on it. It's an antibiotic. We mentioned some antibiotics before, right? So ampicillin acts as an irreversible inhibitor of the enzyme known as? Oh, wow, you've seen that one. It was, by the way, what type of a protease? It's an example of a serine protease, which means what is the catalytic triad? Serine,
And threonine.
threonine, and histidine. Was it threonine?
No, it's I don't know.
Serine and
Is it aspartate?
histidine and not aspartate? Yes, aspartate. I usually say.
Oh, did I just say it wrong? I'm sorry.
Yeah.
This sounds like ASP, wasn't it?
Yeah, you're right. Aspartic acid. No, I just got it, sorry. I was
See, I did it. I drew it wrong
Okay,
in the text.
so that's antibiotic selection in one way. Are there other antibiotics? Another common one used is tetracycline. Totally different mode of action. Tetracycline, some of you know it from acne, right? Okay, so what's its job? It goes in and it binds in reversible nature, right? But it's affecting the site for the tRNA to bind to the ribosome. That's actually what it's doing. It works on both prokaryote, eukaryotic systems, but mammalian systems are less vulnerable. Turns out that bacteria actively pump tetracycline into their cytoplasms, and we don't. So it would be a problem for you, but you don't pump it into your cells, so you don't care. So don't mutate and start pumping it in. Okay, so that's its job. So let's now take a look at this. This actually has ampicillin and tetracycline on the plasmid itself of interest. I cut it. In doing so, note the ampicillin was the green one. I just cut in the middle of that gene. By cutting the middle of it, it no longer would function. But what if it glued back together? It would refunction. Oh, what if I insert my foreign DNA in the middle of it? Then it's not going to function, which is what happens in some of these, but others it didn't happen. Okay, so then did they get put in? To transform, put it in, right? 0 degrees, the bacteria, make it cold. Then incubate it with some salt, calcium chloride, commonly. Rapidly heat it up. We will talk about membranes in the next unit. When I rapidly heat a membrane up, I'm not giving the cell a chance to exchange its fatty acids, which means that the rigid composition of the cell gets too fluid too rapidly, it makes holes. Okay, I literally melted it and it's fluid like butter and it starts falling apart. Okay, and that allows us to incorporate them. In yeast, we use something where we partially degrade the cell membranes. Okay, all right. So you just saw that thrown in. I'm going to throw an agar plate. Agar's made out of what? Seaweed sugars. Okay, cool. Did we learn about that in this unit? It's like forever ago, isn't it? Okay, so we throw this in, and this agar contains tetracycline, which means what?
Bacteria can take it in.
It gets to live if it has what?
The resistance.
If it has the plasmid in it.
Yeah.
Because the plasmid contains the tetracycline resistant gene. So then I take any spot that's up here and I grab a colony and I plate in 2 spots. I take 2 identical gels, okay? They're not identical. This one's got tetracycline just like this one did. And this one's got tetracycline and ampicillin. So I'm gonna spot here, and then the same spot I spot here, I'm gonna spot over here. I'm gonna keep my judicial marks. I know I have a mark here and here. I know what's top, I know what's bottom. And I mark here and then here, it's the same colony. And then I do it again with another colony, another colony. Which ones are the ones I'm interested in given that technique? Explain to somebody else Am I interested in the spot that's growing or the spot that's dead? Tell somebody else why you know which one I'm interested in.
Right. It's stuck in there. Then you know it's the two things. One that die. Not the plasma.
The guy got the plasmid and the
Yeah, exactly. Is this so fair?
Just make you feel like you're back in 181? Really? I literally never, that never happened.
It totally
It never happened.
I know it sounds crazy, but if your buddy was trying to explain to you that you want the dead spot here, because that's the one you're interested in, then the next question, and that is, right, how are you gonna get it?
over there.
I'm gonna go over to the other plate that I have the same colony and I'm gonna sample it. If it's alive over here, I'm not interested in it. Why? It doesn't have the gene of interest in, which should be in the middle of the ampicillin. It has a plasmid, it doesn't have the plasmid with the gene of interest. Now that's crazy talk. Are there other ways to do it? Absolutely. But this is a technique that's used, and it's very powerful to use the two antibiotics that way, okay? All right, so don't get too freaked out about it. It's just a technique, okay, the one that's showing you below. This is a well-characterized plasmid. Like everything about it is known. And they could use it in high school. I mean, okay, it's simple, okay? We got ampicillin, tetracycline, we know how many base pairs it is, PBR322, okay? We've got an origin of replication, we got several different locations where I can use restriction endonucleases to cut them open. We are going to play with that in an activity on Monday. We're gonna cut some up and we're gonna see if you can predict products, okay? It's not all we'll do Monday, we'll finish whatever else we can't finish today. So that's part of the activity we're doing on Monday, okay? Okay, well, that sounds like a challenge, but I got this stuff figured out, right? Okay, are there other things I could use? Could I use something like LAC-Z? Yes. LAC-Z turns the 5-bromo-4-chloral-3-indol beta-galactopyranoside, wow, big word for what? Excal. Most of you would know it as Excal, and it's a sugar. And what does the LAC-Z do? Turns blue if it's functional, white if it's not. In this case, the site that I'm going to add, cut with my restriction endonuclease, put my little piece in the middle, is right in the middle of my LAC-Z. So, if it's not functional, it'll be white. Okay. It's not functional for what reason? What did I throw in the middle of it? My gene of interest, okay? So there are just different techniques that we could use, okay? All right. Once I get my DNA copied and we work it out, can I check to see in the DNA that's created, if I make mobile copies of it, could I see if it was working? Sure, we can actually visualize DNA on a gel. So I can do some gel electrophoresis, and as I put these in, what is the charge of DNA? Negative. Oh, cool. So negative's gonna be, as I put it in, attached to the cathode or the anode? Yeah, well, positive attracts negative, but the anode in this case is what charge?
Positive.
Yeah. So that's what we're doing. So anode means the negative guys are going that way. So that's what we're looking at. So that's what we do. We use a agarose, again, sugar, make a very long 3%, run it on through, do some voltage, and they migrate through. It's kind of a cool little thing. We actually punch holes with a comb right in it and then run it right through with our sample. making this up is like making jelly. If you haven't done it yet in the lab, we even use it in the microwave. We just heat it up, it's long enough, the agarose, polymer, it becomes like jelly, gelatinized. Kinda cool. Okay, so you gotta pour it when it's hot. Okay. Promoter. What's a promoter about? What was that, Nellie?
It helps the proteins that start transcription by
If I want to transcribe a region, it's going to help it recognize the region and start it. And then where's it going to start? Just down from it, it's actually going to have an origin replication, which is really what? AUG. It's the start codon. It starts coding, okay? We've got different operator sequences. We've got the transcription and the termination sequences for these where we can work all this through, okay? So, wow, what do I want to say here? So we are looking right here at a Rec A protein that's been fused to a bacteriophage, okay? And its promoter. Now, under normal growth, none of the Rec A appears, but when the T7 RNA polymerase is introduced, then it will work out. So what I'm actually requiring on is that even with it in there, it doesn't work unless I send along some other cargo which helps it to process the rest of the way. My bacteriophage is delivering more than just the gene of interest, okay? And then it helps create the right protein. So we induce that, okay? This is actually the slide of interest for you in referencing what Lee was talking about before. So now that you're getting ready for studying for an exam and we're talking about all the interesting things you could do, and obviously we could go on for a very long time here. This one tells you benefits and problems, benefits and problems. If I were looking for a slide to study, this would be the one. Why? Because I'm summarizing. Why would I use a bacteria plasmid? Well, it's going to be well characterized, easy to use, scalable. I feed it sugar, it grows. Protein folding is going to possibly be a problem if I've got a eukaryotic system and I put it in a bacteria. Proteins with covalent modifications probably won't recognize it and put the modifications on. Proteins that may aggregate because they really needed a chaperone. Oh, okay. Bacteriophage with a bacterial phage is a virus. So it's a virus that infects the bacteria. Can I put this in and get more information? Sure. I could be able to use the RNA polymerase rather than the host that I just barely talked about, okay? High levels of gene expression, efficient gene transfer, this all looks good. Still have problems with these things, okay? Yeast, we talked about some advantages of yeast. Again, eukaryotic, proteins more likely to scalable, where characterized, but it's hard to get the DNA. It really is, you guys. If you ever try to break yeast cells open, they're tough. Okay. In my graduate work, one of my guys was working with yeast. Man, he tried like every technique he could do to rip that stuff open, figure out which one was the best. None of them are fantastic. Protein folding, modifications are still potentially an issue. The more I come this way in higher eukaryotic systems, the more it's going to be favorable to making it work, okay? But it's also going to get me less and less. It's going to be more expensive and I'm going to get less protein. So if I can make it work in any way in the simpler system, life is easy. That makes sense to everybody so far? So that's your list to look at for selecting, okay? Why would I be interested in doing this on a high level description without going into all the details of memorizing anything, because that would be a waste of your time because the palette is big. Could I make other molecules like pigments, antibiotics, drugs, surfactants, flavors inside of bacteria. Or other small plants, or, yeah, I could put these things in. Could I do things like alternative fuels this way? Absolutely. So the more that I wanna play with, what could I do with my organic chemistry reaction in a biological system where I'm literally just feeding it sugar and extracting my molecule of interest, the more and more beautiful it looks. In some sense, we call it a world of green chemistry because it looks a lot more like God. Can bacteria produce poison?
Yes.
Yeast produce poison. Yes, everything I throw could produce a toxin. But if I'm designing it to produce just what I'm interested in, life is good. There are some molecules you can put in that you don't have to kill the cells in order to harvest the product. It will actually expel them. That's cool. Others, you have to slaughter the cells, break them all open to get the product out and purify it. If you can get one where you can keep the cells alive and just have it elute, that's really nice. So that's part of the secret is, could I put that in? If I need a transporter, could I put that system in too? What are the things I can do? Okay, so there's some things that we do with it. Here's one of the ones that's perhaps most famous for all of you. A few years ago, they had a big brouhaha. The government got involved in this issue. Turns out that years ago, how did we get insulin to save those with type 1 diabetes? We killed pigs.
Yeah.
Wait, we were killing them anyway. We were. And where were the pigs going? To bacon and ham. Pork chops, right? But that was a slaughterhouse. And then we grabbed their pancreas and we try to squeegee out all the insulin we could get out of it, hoping that the candidate didn't get what? If I now inject this right into your blood. Anything else that might be a contaminant when you're working with raw pork. Oh, and it was expensive. And I had to kill animals for it. And so it wasn't what we call a scalable solution. So somebody came along and said, I think I can put this in a bacteria and make it work. But what was it? A eukaryotic system. It's one like you have for insulin that's got to be in here. And so we worked on creating that. So now we feed it sugar and it kicks it out. But what was happening? Pharmaceutical companies who'd invested all that technology to do that made it really expensive. But how expensive was it once the technology had been around for a few years? They're feeding bacteria sugar. It's really cheap. And then they're purifying it. It's really cheap. And so the government got after them and said, you guys, why are you making all these people suffer with these expensive things? And some people are dying because they can't afford their medicine. Make this cheaper. So they stepped in. Monopolies are hard. There can be corruption. We don't need to go into the politics of it. But there is invested technology here too. So realize there's a win and a lose, right? Okay. Are other systems worked on? Absolutely. Yeah, a lot. One of the ones that I talked about in the next one, we make things like polyester in bacteria. What do you use polyester for? Clothing. You can make bacteria make material? Absolutely.
Cool.
Polystyrene, woo! We're actually designing bacteria now to actually create enzymes that will break down plastics in the middle of the ocean. Hi, that sounds like a great idea. We're working on all these things. Okay, so that gives you some idea of why we might be interested in doing this type of work, right? And could this be your career? Yeah, in a world that's ever more and more filled with pollutants, this is a good idea. Okay. So, get on the bandwagon, make yourself rich, start your own company. Life is great, right? Site-directed mutagenesis. So, I'm going to now create a mutant where I'm going to replace one or two of the amino acids in key locations. Have you seen that in some of the papers that you looked at? Yeah, absolutely. So how did they do it? Okay, so I've got a function of a protein, I wanna knock out a certain protein, that's one level, but what if I wanna keep the protein but just affect the few amino acids? That's a whole other story, okay? So this is how we go after that. You gotta know the specific importance of the amino acid, so location's a big deal. I know that you're gonna make your hemoglobin be super hemoglobin, so you can like go up, fly and everything else, that'd be awesome. But where's the mutation gonna happen? How do I get this into the interesting plasmid? Once I put this in the plasmid, then I can transform it. What am I transforming into? Double-stranded DNA? Oh, okay. Okay. And then we can actually purify that. If I do this just right, how do I fish out my proteins of interest? Well, we can put on it a poly A tail, a his tail. We can go after it. We can put on things like maltose binding, galactidase. There is ways to purify with chromatography or with beads our proteins of interest if we just put a little bit of more mutation on the other protein. That makes it easy to fish them out. Remember our how do we purify proteins? There's the game. So we can actually mutate it along the same path, make it easy to get to. And then we go to work and we purify it. Okay. A cDNA library. Wouldn't that be cool to have one of those? So if I know that I have a messenger RNA that's producing an interesting little thing, maybe I could get, messenger RNA is how stable? Not very stable. Could I make it into DNA instead that would be very easily stored, more permanent for me? Absolutely. So I actually want to go backwards. I want to use a reverse transcriptase. I want to actually get this into, at first, single-stranded DNA, and then eventually double-stranded DNA. And that's the approach that's being looked at, okay? So we start with a primer that recognizes that poly A tail, and then we go backwards on that, okay? Other things that we might use to find where the protein we just put in a cell, where is it going? GFP, have any of you guys used GFP to find where something goes in a cell? Look at it in an electron microscope, oh, look at that, that's where it is. It's pretty cool. Use that in my research too for that reason. GFP is easy to find stuff, right? So, immunofluorescence, another focus on that. So antibodies, putting that way. Here you see some nice worm that we put it in. Did you know that we spent with our government, because it was a good idea, that after we discovered GFP, which was naturally there, we decided we wanted more colors. Some researcher spent more than a decade of his life creating the rainbow. And millions of dollars of your taxpayer money.
Just to get different colors on it.
And it works cool. It's so awesome.
That's true.
And we play with it, and it's really awesome.
Can't doubt science.
What's so fascinating to me about GFP is a beta barrel structure, right? Structure motif, is that we throw that little lead weight on the end of some small protein, and it still seems to operate for the most part and go to the right location.
Wow.
You just got to think about it. It's not supposed to have this, and we just added this big tail so we could find where it was, and it's massive. And it's saying, well, I know how to get to where I'm supposed to go anyway, and it gets there.
Wow.
Is that true all the time? No, it's not. So some researchers overuse it, and they make assumptions that are incorrect. Most recent research that I just looked at, last week I think, we had a researcher come in and he actually was pulling off in beta barrel structures. He was actually making it so that when we did ELISA assays, that they actually had two different proteins that when you bound with the antibodies to the right locations, then the fluorescent molecule at GFP would be made. Because it would bind, and then it was close proximity, and then it would actually finish the protein folding to make a fluorophore. Clever. And the beauty of it was, instead of ELISA assays having all the washing steps, there were no washing steps. That's probably way of the future on that sort of thing, but not my work. But there you see some antibodies using immunofluorescence. You get it, fantastic, okay? All right. Yep, interesting. PCR, probably ought to know that, right? Because that was on the study guide. Remind your neighbor of what I said on Wednesday. Is PCR, which stands for polymerase chain reaction, is it making a copy or is it sequencing DNA? Tell somebody else the answer to that question.
I think it just copies it. It just makes it. Doesn't it say obviously it's just polymerase chain reaction? So over and over again. Really? A bird. But based on the copy. It's a bird. It's a plane.
Nice try though.
Okay, if your neighbor said that it's only making copies and you believed them, you're probably in good shape. You'll probably get something right on the exam. If you think it's sequencing, you're wrong. But I do need to know where to start it and where to stop it, which was part of our conversation prior to class. And that means I need a primer region, but it doesn't mean I know the sequence. Okay? And so we're going to amplify and make them. Okay, so what we need is something called a thermal cycler, which is just a little incubator that gets hot and cold, hot and cold. How hot? About 95 degrees. How cool we get? 50 to 60. Okay, we're going to talk about this. So when we talk about it, every time I make a copy, I just keep increasing this about 2 to the nth fold. So after 20 cycles, to the 20th. Whew, that's cool. That gets me a large amplification, about 1,000,000 folds. So let's see what this looks like. This is the region I'm interested in. These are the primers. Notice the primers are on the opposite strands. 3' to 5', 5' to 3'. These are going to copy which directions? The same way always that polymerase works. 5' to 3'. So it's going off the template strand and it's going off the 3' to 5', but it's writing 5' to 3' every time. So let's see if you can follow this little clip and make sense out of it.
PCR uses repeated cycles of heating and cooling to make many copies
Heat
of a
it up.
specific region of DNA.
DNA is going to do what?
First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands.
Now, at this temperature, what's going to happen to most proteins?
Nature.
Denature and be destroyed. So we found a special polymerase where? Yellowstone. Okay.
When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand.
Means there's a whole bunch
This
of
completes the
nucleotides
first
just hanging
cycle.
around in that solution.
In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers
This
begins
is the
to
first
accumulate.
time that I got the short sequence I'm interested
After
in.
30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
After we get those short ones, those are the ones that become the predominant strand.
Dang.
Okay, and yes, after 30 cycles, we're up to 1 billion. Okay, so that sounded interesting.
Hello, everybody.
Yes, so fun. When we were doing that, when does the primer add? In the hot temperature or the cold?
Warmish.
I open the DNA up in what? Hot. And then I cool it down to 50 to 60. We still feel like that's hot because that's uncomfortable for you and I, but in relative comparison, it's cold. That's when the primer adds. Yeah, it gets a little bit warmer for Taq polymerase to work, but Taq polymerase working in hot or cold? Medium, but really cold in reality. Because if it was hot, what would happen to it? The DNA would separate again. It wouldn't be any value to me. So I'm working with the primer and the Taq polymerase at relatively cool temperatures in comparison to the when I'm opening up so I can make a new copy. Okay? So just understand that, you might be able to get somewhere. Okay. Are there other fun questions in the back of the book? What do you think, Lee?
Yeah.
Okay, sounds awesome. Cool. Maybe it works out. Okay, DNA fingerprinting. Did you know there are spots in your DNA that are actually well-studied enough that I could fingerprint 13 well-studied locations, and actually I can determine whether it's you or somebody else with fidelity of 1 in 10 to the 18th. Meaning, what are the statistical odds that I'm gonna make a mismatch and say you're the guilty one and you go to jail? Very low. Scientists can now get it at 26 hours and $600.
Dang.
That was the target. That's what we wanted. Do it in a day, do it for under $1,000. So, how much has been the economic impact of going to the moon? Fantastic. Well, what about the Human Genome Project? Well, we invested in reality $3.8 billion, but it's given us $796 billion as of 2012 in economic impact. That's the jobs it's kicking out. 310,000 jobs, and now we're curing diseases. Probably worth our taxpayer dollars. But not all science pays off that way. But this one, it's a good idea, okay? Okay. We already talked about next-gen sequencing. We already talked about that pyro sequencing element of it, that when it binds, it actually flashes for us. So we talked about that and talked about how that works. So that's all cool. So in the end, it mentions this idea of personalized medicine, okay? So a couple of things in the last minute as we try to finish where we're at, okay? There's a lot of other things we could talk about. The dream is due to the over $1 trillion of spent money from our government into research to identify biomarkers, which was part of the research that I did. We just create biomarkers for diseases. Turns out that most diseases, you can't identify that disease by one marker. You actually need like 15 or sometimes 20 markers to actually characterize that's this disease versus that's this disease, because the differences are subtle. Diabetes happens to be one of those, okay? So we spend a lot of money identifying biomarkers. Everybody published their papers and they're sitting out there in the archives ready to be mined and validated. And if they're validated and they go to market, then what could happen to the doctor who's in the office that has the ability to test all those biomarkers and say, if this is up and this is down, and this is up and down, and this up, then I'm gonna actually say this is this disease. What if I have, of the 20, I have 15 that are on in the right direction? Well, then I'm well underway to getting that disease, okay? So the idea is, if I could identify the right biomarkers in the right set, maybe I could actually diagnose diseases early, which would mean we could get into preventative medicine instead of just treatment. Right now, people in diabetes essentially walk around and say, hey, I'm obese, but I've been obese all my life. And they exist that way for sometimes 20, 30 years, and then overnight, the doctor says, guess what? The reason you're having problems and you're urinating all the time is because your kidneys are shot and you have diabetes. Okay? and you'll be blind in probably the next 20 years. Congratulations. Here's your new life. And people go, what? I just literally had a heart attack and my life has changed overnight. It's really what it feels like for a lot of people, okay? So could we get certain diseases we could diagnose better? We could watch them earlier, and the answer is yes. And we actually did the research on most of these diseases already. So what would personalized medicine look like? Validate the markers that are already there, make sure they work, and then take them to the clinic. How would we do that? Do you remember I mentioned something called an aptamer? Now an aptamer is a strand of DNA that's short, like RNA, but it folds up and it's got high affinity for just one specific ligand. It's like an antibody, only it's made out of DNA. We can fix those to little plates, and just like I'm going to show you in just a minute how we can do sequencing for SNPs, you can actually lay down about 300,000 of those in a plate, and actually I can know, based on a fluorescent signal in probably less than 3 minutes, because that's how it works in DNA, which ones are up, which ones are down, and I can process a report that would tell you what actually matters. Now, if you went every time to the doctor and you did a little pinprick, I could give you that report. Imagine that you went to the doctor, you moved out of the waiting room into the office, you sat there just like you always do, and the nurse came by and pricked your finger, and then less than 10 minutes later, because you already waited that long anyway, the doctor walked in with a chart and he was actually talking about the things that were wrong with you. That would be brilliant. And that would be personalized, especially if you also had with it your DNA that he'd been watching his whole lifetime to say you're predisposition to this and this and this and this disease. That's what risk of it. You still have to insure me. Oh, that was a smart move. That's the problem. And currently the problem is insurance companies versus you. Who owns your DNA? That's the one that hasn't been decided yet. Insurance companies, they think they own. All right. How much of your DNA is protein coding? 1.5%. Wow. Look how much is introns, by the way. Yeah. So here's what I was talking about with this, getting a SNP, a single nucleotide polymorphism. This is a gene chip. That's what your aptamer array would look like. By the way, this is little DNA strands, and we throw it at it, and we can actually identify all the ones. Okay, so there's aptamer arrays right there for you, and yeah, there's personalized medicine, all the things. Did you know, by the way, as we leave today, that there are drugs which we know won't work for a patient, and there are drugs we know that they will, and we can even give you dosage amounts based on if we knew your own DNA. That's awesome. But how many people's DNA do we know? Not none. That's fascinating, though. So could we do better? Is it possible that you need to be eating high leafy greens and somebody else needs to be eating more iron in their diet?
Oh, yeah.
Yeah, highly likely. So we need to get better at this game. So we can actually prevent disease, not just treat symptoms. So that's the goal of personalized medicine. We move on our way. And we'll talk about all the awesome things of CRISPR, but that's all not on the test. So come back on Monday.